Okay, thank you. First of all, I'd like to thank the organisers for inviting me to speak today. So I'm from the Ellis Lab, which is part of the Centre for Synthetic Biology and Innovation at Imperial College, and I'm going to talk to you today about some of the work we've been doing with yeast, and specifically our work with synthetic yeast genomes. So I'm going to give you a quick overview of yeast. I love yeast, and hopefully by the end of this talk, I'll have persuaded you to share some of my passion. So when I say yeast, I specifically am talking about Saccharomyces cerevisiae, and this is what is commonly known as brewer's or baker's yeast. It's a single-celled fungus, and we actually understand the biology and genome, or at least we think we do, comparatively well. And both as a product of and because of this, it's a very important organism, both for basic research and particularly in biotech. And as some of you may be aware, it is the greatest organism on Earth. So it's often described as the original biotechnology. In the region of 10,000 years, people have been using yeast to produce food, whether that's through baking, winemaking, or brewing. And the main reason for this that everybody is taught at school is that yeast performs a fermentation where it takes glucose and it converts it into ethanol via the release of CO2. And this is obviously a very handy process if you want to make alcoholic drinks or release CO2 into your bread dough to make it rise. But actually what's happening in yeast is a lot more complicated than this. So here's just a quick slide that I stole from a review that shows you some of the flavor compounds that are released by yeast during a typical fermentation. And here's just sort of a brief overview. And what's worth noting is that different strains will produce all these different compounds in different amounts. So depending on how you want your beer to taste, you might want a very specific strain which moves everything towards a particular metabolic product. So for this reason, being able to generate a diverse number or isolate a diverse number of strains for your particular application is important in food and drink, but it's also very important in various other biotechnology applications. So yeast is very important, for example, for the bulk production of ethanol, for bioethanol as a fuel, and it's also a very frequently used factory for recombinant proteins, pharmaceuticals, and fine chemicals. So to a degree, these needs can be met by mining the genetic diversity found in nature and in strains that have grown up around certain old breweries and things. But in order to really get the most out of your yeast cell for bioproduction, a certain amount of strain engineering is often needed. And the most classic recent example of that, I guess, is the work by Kiesling and Co. to get a yeast cell to produce artemisinic acid, which is a precursor of the antimalarial artemisinin. So this is very well publicized, and it was seen as a great victory for synthetic biology, and it displays what can be done in terms of strain engineering in yeast. But it's worth noticing that the process took 150 person years and cost around $25 million. So although the technology to do amazing things is there, it's not easy, fast, or cheap. And this brings me on to SC2, which is our project to generate a synthetic yeast genome to try and address this. So when I talk about synthetic genomes, what I'm talking about is a genome that is working within a cell that consists of DNA that has been designed, typically in silico, so on a computer, and then actually chemically synthesized before being transplanted into the cell. So rather than having a physical precursor in an existing organism, this is stuff that we've designed and chemically printed out. So although the sequence itself may derive, the information may derive from natural sources, the physical flow-through of the DNA is that we have designed and then chemically synthesized it. So why we want to build one of these? Well, the ability to go from an in silico genome to hopefully a functioning genome in the cell really opens up the opportunities for what we can do with an organism. And really importantly, it means that we're not as limited by what already exists in nature. So it's actually quite a good academic exercise in of itself, because by building a genome, we can hopefully help understand how natural genomes work and also test out whether our theories on how those genomes work are actually true. If we build a genome based on our principles and it doesn't work, we know that there's obviously something wrong with our understanding of the genome. But certainly more in the context of today, what building a synthetic genome allows us to do is enhance these genomes in ways that aren't currently easy or even possible. And this is a particularly useful tool when it comes to strain improvement for biotech. So, the synthetic yeast genome project that is running at the minute, rather than copying the yeast genome as is, we're actually designing in some very specialized functions so that the cell that we get out of the other side can do things that natural organisms can't. So these are the various things we're doing. Don't worry too much about the details. They can be bundled into a few categories, basically. We're deleting and moving certain elements, and that will help us improve the genomic stability of our strain. We're also recoding the various things that will help us identify synthetic strains, and down the line will hopefully help us improve biosecurity and allow us to introduce new protein biochemistry into the cell. But what I'm particularly going to talk to you about today is the design changes we've made, which will allow us to induce rapid evolution within our synthetic strains. So the project to generate this is quite large in scale. The yeast genome is around 12 million base pairs, just over, and it's split over 16 chromosomes. So this is quite a sizable amount of genome to design and synthesize. And for this reason, this is actually being done by a large international collaboration. So there are labs all over the world, in the US where the project originated, as well as a number of labs in China, Singapore, Australia, and 2 labs in the UK are all building a chromosome each. So for example, at Imperial College, we're building synthetic chromosome 11 here. So what will happen is all these different labs will each build their chromosome, and then once they're ready, they will go together into a single yeast cell, and you will have a yeast that is entirely powered by a synthetically designed and synthesized genome. And the progress for this is going very well. In terms of the literature, currently the only published completed chromosome is chromosome 3 here, which was reported in 2014. But there are a number of publications imminent of further chromosomes. And this is the progress of the various chromosomes as of December. But I can report that since then, within the last month at Imperial College, we've actually finished completing our synthetic chromosome 11 towards this project. And I built this with lots of help from Maureen Dreesen, who's our fantastic technician in the lab, and really put in the work with integrating synthetic DNA into yeast. Okay, so that's an overview of what the synthetic yeast genome project is, briefly. I'm going to go now into how we're going to use this, hopefully, for biotech applications. The system I talked about for inducible rapid evolution is a system called Scramble. Don't worry too much about the complicated name. It was basically generated so we could go for the acronym Scramble. But what it basically consists of is that when we designed our synthetic genome, after every single non-essential gene, we inserted a special DNA sequence. And this is called a LOXP-SIM recombination site. So what happens with these sites is they can be bound by a protein called recombinase. It will bind 2 LOXP-SIM sites and cause a recombination event between them, essentially causing a genomic rearrangement. Because we have this site after every single non-essential gene, this opens up a huge number of possibilities, because what we can do is we can express this recombinase protein that causes the recombination on a plasmid within the cell, and we fuse it to an estradiol binding domain. So what this binding domain does is it prevents the recombinase from entering into the nucleus. So for the most part, this protein is just hanging around in the cytosol and able to get to the DNA within the cell. If we add a chemical inducer, in this case beta-estradiol, it will bind to the binding domain here, and this causes a conformational change which allows the recombinase to be imported into the nucleus. And when these recombinase molecules enter the nucleus, they can grab any 2 of these LOXP recombination sites, and these are symmetrical sequences as well, so they can bind them in either orientation. And what they then do is flip them over and cause a recombination event. And these can lead to deletions, translocations, duplications, or inversions within the cell. And what's important to realize here is this isn't a single event. There are lots of recombinase molecules, and they're causing lots of these recombination events. So what you essentially have is a real large-scale shuffling of our synthetic genome sequence. If you think about it as a pack of cards where each card is a gene within the genome, you can basically shuffle away as much as you want, but rather than just shuffling the genome around, you can also copy new cards and add them in, and you can also throw cards away. So the amount of diversity that can be produced in one of these experiments, the number of potential outcomes, for all intents and purposes, tends to infinity. So last year, a paper was released where they did this scrambling reaction on a small section of synthetic genome. So this is a strain where only 100 kb, basically one chromosomal arm, was in the synthetic format. They scrambled a parental strain, and then they sequenced a number of colonies that came out the other side to see what happened to its genome. So I don't know if you can see, it's quite small, and is apparently right up the top there, above the screen. But these two here are the parental strains. Each arrow represents a region between two of these recombination sites. The arrow's orientation is the orientation of the gene, and they've given a color code just to give an indication of where that particular section sat in the parent genome. And just by randomly selecting things that have been through this scramble sequence, you can see that they've isolated cells that have a huge diversity in both the size of that particular region of the genome and also the orientation and order of the genes within there. So this is a nice demonstration that even when you're only doing this to a very small amount of sequence, you can create huge diversity. So this got us thinking, this is very nice as an academic exercise, but how about if we did this and in combination added a selective pressure to basically turn this into a souped-up directed evolution? The idea being you have your synthetic cells, you add your chemical inducer, which causes the genomes to scramble in this way, and then you add a selective pressure and try and select individuals out that flourish under that particular selective pressure that represents your condition of interest. So we decided to do this. At the time that we did this, our synthetic strain was about a third completed, that chromosome. So we did this with a cell in which chromosome 11 was a third synthetic, so formatted with these recombination sites. We then set up two cultures, both of which had the synthetic chromosome section here, but one was expressing the CRI recombinase and one wasn't. So basically these on the left can scramble and these can't. In a single day, we grew these up for 4 hours and then added chemical inducer to both for 4 hours and then washed and plated and incubated these plates at 37 degrees. And what happened was really exciting for us, basically. We found that the cells that couldn't scramble because they lacked the recombinase looked as you would expect cells would after being incubated at 37 degrees. Yeast cells don't like 37 degrees. Their ideal growth temperature is 30 degrees, so this represents a bit of a heat shock situation for them. And the cells that didn't scramble, they looked uniform and they looked kind of not particularly happy, but generally okay. With the cells that had scrambled, first off, 90% of them had died because they'd flipped out an essential gene. But of the ones that remained, a lot looked sick and quite weak, but there were some individuals that had formed absolutely gigantic colonies in just 2 days. So we streaked these on and grew them overnight at 37 degrees, and after one night at 37 degrees, we saw this. So the unscrambled cells, there were barely pinprick colonies, which is what you would expect from overnight growth from yeast. But the cells that we identified in this single day's worth of scrambling grew absolutely huge colonies overnight, which is quite a dramatic phenotypic change for yeast, and we've not seen any evidence in the literature, or at least I haven't found any, of yeast cells growing this fast overnight on a plate. So this is really exciting. This is just us trying something out quickly with what happened to be present within that small section of our chromosome. So we started thinking, what happens if we build the expression of heterologous pathways into this? So this is introducing foreign pathways from other organisms and seeing if we can also enhance the phenotypes for these. As long as we have a selectable condition, such as there being a colored product or inhibiting bacterial growth, for example, or growth on an unusual carbon source that yeast can't normally metabolize, then we could theoretically use this scramble system to quickly find optimal strains for production of our particular molecule of interest. So we actually got a master student, Jack Ho, onto this. And what we got him to do first was to see if he could get a strain optimized for growth in xylose. So xylose is a carbon source that yeast can't usually utilize, but it's of interest because it's very prevalent in waste biomass. And if you could get yeast in your biotechnology applications to grow on xylose rather than glucose, not only would it be cheaper because you're using a waste biomass, it would also cause reduced competition to food resources. So what he did is he took a plasmid and he encoded on it the genes that yeast needs in order to utilize xylose. What's important about this is the pathway itself wasn't formatted with these recombination sites. So during our scrambling system, all that's happening is the background strain is changing, the pathway itself isn't. Okay? So when he had a strain with no plasmid, he saw no growth in xylose. When he put the plasmid in, which allowed the yeast to utilize the xylose, we saw some modest growth, which is kind of similar to what other people have seen with these methods. And when he scrambled and tried to competitively select for a strain that was growing particularly well after the scramble in xylose, he managed to find a strain that growed very, very well indeed in xylose and was utilizing it very efficiently. To put this into the context of growth rates here in these bar chart here, so no plasmid, you see basically no growth. The unscrambled strain, so this is just the plasmid, you get a modest amount and absolutely huge amounts with our scrambled strain that we managed to isolate. To put this into perspective, this is growing around half as fast as yeast normally does when it's using glucose as a carbon source. So that's it's a huge optimization of what we were previously seeing. So on the back of this, we decided to see if we could get him to look at Scramble to optimize some other things as well. So we're kind of interested in violacin in the lab. It's an antibacterial compound, and it's been earmarked as a potential cancer therapeutic. And very handily for our purposes, it turns the cells a purple-black color, so it's actually quite easy to select for colonies that are producing it better than others. So this is sort of a perfect case study for our system. So using the Golden Gate kit within the lab, we assembled together this 16 kb plasmid, which expressed all the genes needed to produce violacin from a tryptophan. And he did this scrambling system again, and this time he grew up different colonies that he picked and just spotted them onto the plate so that we could try and isolate colonies that looked like they were producing more violacin than others. And sure enough, you can see there's a huge amount of diversity from all the different colonies that have gone through this scramble because they've all had different events happen within their genomes. And he isolated this colony here, VB2, V for violacin, B2 because it's B2 on this grid. And this looked to be producing violacin particularly strongly. And sure enough, when we did this with a more quantitative colorimetric assay, it produced twice as much violacin as the strain did prior to the scramble. What was particularly interesting about this strain, though, is we decided to use it to look at how this was happening. We figured out one way that it could be improving expression was generically boosting production from a 2-micron plasmid. So these pathways are present on a particular plasmid which has the origin 2 micron. So we decided to take this plasmid and rather than the violacin plasmid pathway, just put a GFP marker on there. And sure enough, this is the output from the GFP. You get if there's no GFP plasmid, there's no GFP, obviously. If you just transform in the plasmid, you get a decent amount of expression. But again, you get around twice as much if you transplant this plasmid into our strain that we isolated for enhanced violacin production. So it looks like this strain is universally increasing expression from any pathway that you put on this particular plasmid backbone. So we decided to see how far we can push this. Along with another postdoc in our lab, Ali Owan, we basically spent 3 or 4 years trying to get yeast to produce penicillin-based antibiotics that are bioactive. And this is a very complicated process. We eventually got it to work, but it involves non-ribosomal peptide synthetases. It involves peroxisome targeting of certain enzymes in the pathway, very in-depth pathway design, and kind of the cutting edge of library-based DNA assembly to create all these various ways that we can try and enhance our production of penicillin. And we were aiming for a strain that could generate enough penicillin in its supernatant for that to be bioactive against Staph aureus, which we eventually managed to do, but it took very experienced people about 4 years to get to this place. So we took the pathway, we transplanted it into the strain that it had taken the master student one day to generate, and sure enough, he doubled the expression of the pathway we'd been banging our heads against the wall to try and optimize for 4 years. So I think this is a very good indicator of just how powerful this methodology is. So yeah, we see SC2 as a potentially really useful tool for biotechnology. Even with just the Scramble element of the genome, we can show that it generates huge diversity within a pool of cells. And if you do this in a test tube, you have millions upon millions of these cells that you can potentially select from. And doing this allows selection of enhanced phenotypes. And when you tie this in with adding heterologous genes and synthetic pathways as well, it's a really powerful way of optimizing your biotechnological application. And most importantly, the process is incredibly fast, incredibly easy, and incredibly cheap. So if anybody's particularly interested in this, we have a website, syntheticyeastresource.com, which was made by Rob in our lab. And here there's progress updates as to how all the various different chromosomes are getting on. There's also a genome browser where you can browse all the sequences. There's a methods repository. And also, once the strains become available upon publication, this will act as a strain repository as well, where you can go on and request strains to be sent to you. And these strains will be available for use for everyone once they are published. Okay, so in summary, SC2 is a big international collaboration, and we're creating the world's first synthetic eukaryotic genome. And we're making lots of changes and learning lots as we go. But we're also, including all of these new design features as well. And we've shown that scrambling synthetic chromosomes, yet alone genomes, can generate huge diversity and allow for the selection of enhanced phenotypes. And once again, this will be freely available once published. So I'd like to thank the organisers once again for inviting me to talk here. Thank you for listening, and thank all the people who made this work possible as well, particularly Maureen Dreesen, who was my co-builder on the chromosome, Tom Ellis, who runs the Ellis Lab at Imperial College, and all our various international collaborators as part of the SC2 consortium. And the funding as well was from the BBSRC and the EPSRC. Thank you very much. Okay, any questions? That's right. So, yeah, I mean, I didn't really have time to go into it too much. I'm glad you spotted it, because it makes a good question. So what happened is when we were looking for potential phenotypes to look for when we scrambled yeast, obviously we were working with a limited amount of the chromosome that we had synthetically formatted with the recombination sites. So yeast has a great community behind it. So things like the Saccharomyces genome database has a lot of functions, including this yeast mine, which allows you to mine a huge amount of phenotypic and transcriptomic data that people have generated over the years. So what you can actually do is simply ask YeastMine, okay, these are the genes I'm interested in, which phenotypes are associated with overexpression or deletion or knockdown of these genes. And one of the phenotypes that came up for quite a few of them in the region we looked at was response to heat shock. So that immediately put us on to growth at 37 as being a potentially good one to look at as a proof of concept for this, because we were reasonably sure we'd get a result from it.
Stable are your comments, so to speak? Or do you have to treat the recombination machinery? You rearrange everything.
Yeah.
Is it going to become unstable?
In theory, no, and we've not seen it be unstable. Although the, where are we? Here we go. So this plasmid here is not typically in the strains, okay? When we want to scramble, we transform it in, we scramble, and then we cure the strain of this plasmid, so the recombinase is no longer present. The actual recombination sites themselves are 34 base pairs in length, and that falls below the threshold of what the yeast native recombination machinery will recognize and used for homologous recombination. So these sites as of themselves are not sufficient to cause recombination events within the cell when Cre recombinase is not present.
Do you have any insight into the molecular regulation? Do you say the promoters are still the same for all the expression of the penicillin enzymes? Like, are there any idea of what you
We actually don't. We have no idea. Or basically, all we see is that, we measure specifically for ACV, which is a product of the non-ribosomal peptide synthetase, and benzyl penicillin, and we basically just see a doubling of both. The thing about Scramble is it's kind of a black box situation for the most part. We asked the Scramble a question, and it gives us a strain which is better at that. If we want to actually pull apart what's happening within the cell, that's a lot more complicated. So we need to start doing things like whole genome sequencing to see which regions have been deleted and duplicated or have expression changed. So, which is doable. We're working at the minute with nanopore sequencing to try and get a pipeline for rapid deconvolution of what happens in these scrambled strains. But for the most part, it's actually quite tough to say what is causing these phenotypes. We just know that we can generate lots of strains that give them to us.
So I'm just wondering, have you used the scrambling system for optimizing recombinant protein production? And how do you select, in that case, growth or color changes?
Sure, absolutely. We haven't so far. This is still very much the early days, and these were just some fast examples we were able to do. But I agree. It's the same with directed evolution, right? You're only as good as your screen. What we found encouraging, though, is with the violacin here, we were able to get a strain that generally improves expression from a particular vector system. So with this, basically, just by selecting a specific vector system and selecting this strain, we're able to double general production of whatever heterologous gene or pathway we express in this way. So hopefully there will be other examples of universally good production strains. The 2 micron, ooh, good question. It's a relatively high copy number. It's about the highest copy number we have for yeast. So presumably what's happening here is that there is some sort of copy number regulation that is changing.
Yes, with the genome itself, or obviously we have a lot more of the actual construct. They have a high number of copies of the construct that the genome itself on it.
So these aren't on the genome, they're on a separate plasmid, and the plasmid itself can't scramble. So one potential thing that can be happening here is the cell's own mechanisms for regulating plasmid copy number are being changed somehow. So we're actually messing with what the cell thinks is a good number of this plasmid to have in the cell.
Do you think this completely applies to other strains of yeast, such as Saccharomyces?
Yeah, I mean, the synthetic yeast project itself has taken a long time, but both because of background factors with technology increasing and also the technologies that have come out of this project itself to try and make this a viable option. If you were to make another genome now, it would take you a fraction of that time. So we foresee there will be lots of other synthetic genomes built, and POMB is obviously a good candidate because its similarities in the cell cycle to the human cell. Okay, thank you very much.